All right, so first let me describe the population. As Giala mentioned, we used EKW outbred population from the vendor. It has been maintained by mass breeding for about 12 generations in the Sugio's lab. From this population, we selected some breeders, and these breeders generated more than 6,000 larvae for behavioral tests. So I need to mention that we deep sequenced a subset of these breeders, and it's important. It will be important in a minute. For the larva, we developed this method, which I will describe, which I'm describing. And we also deep sequenced a small set of larvae to generate a truth set, which we use for measuring concordance. And this is our sequencing method. We isolate DNA using a high-throughput 96 batches bit-based kit. Then we prepare a library using Riptide protocol. Riptide protocol uses random priming from both ends, and it also can be done in 96 samples per library, very high throughput. Then we sequence the libraries in the batches of 10 libraries per lane, and it derives 0.5x mean coverage. For bioinformatic part, we align it to the genome assembly available from GRC. Then we genotype, then we call genotypes using Stitch software and using these 87 deep sequence breeders as a reference. We only use Stitch for iteration 1, and then we impute genotypes to reference using Beagle. This process, and then we measure accuracy by comparing genotypes with deep sequenced individuals. So this process we call 10 million SNPs with high accuracy. I wanted to point out one caveat of this process. The libraries are prepared in the batches of 96 samples. So within the library, we get a wide range of number of reads, and the individuals with a low number of reads have too few reads to call genotypes confidently. So we end up needing to redo from DNA step, from library preparation step, approximately 5% of samples. So this is kind of caveat of this otherwise very low cost and very high throughput process. As you might remember, we sequenced only a subset of breeders. So the question here was, does this subset represent the haplotypes in the population well enough? In other words, can we use just a subset as a reference to confidently call genotypes in all larvae? And this is the experiment which we did to answer this question. We excluded one of the pairs of the breeders completely, and then called genotypes with and without this pair. So here you can see concordance between genotypes when called two different ways. And the individuals in red are the individuals who don't have both parents. So both parents are excluded from genotyping, from the reference set. In blue, only father is present. In green, only mother is present. So as you can see, the excluding specific pair of parents does not really affect the accuracy of genotyping. So this shows that our reference set is sufficient. And finally, I wanted to show you another cool thing, how we use genotyping data to validate and correct pedigree records. It turned out that approximately 10% of samples where pedigree was recorded, it was incorrect. And this has to do with the mechanics of the experiment, how eggs are collected, how larvae are transferred. They just got stuck to the tank and to the net. Since we have genotypes for both parents and offspring, we can correct these genotyping errors. So to do this, we measure Mendelian error rate in each individual. So in this graph, each column is one family. And on y-axis, it is Mendelian error rate. So you can see that in the green box, siblings in each family has very low Mendelian error rate. So this is a good pedigree. But in some families, there are siblings. I also need to mention that only problematic families are shown here. There are multiple families which are not problematic. So this is just a subset of problematic families. So some siblings actually have high Mendelian error rate. And this means that they were misassigned to the family. And since we know genotypes, we can assign them to correct family. There is also a set of families where all siblings have a bad Mendelian error rate. And this means that the breeder was misassigned. And again, since we know genotypes, we can correct this error. In conclusion, we developed a low-cost, high-throughput methodology for genotyping zebrafish. It produced death genotyping with high accuracy, and this method enables the first GWAS in outbred zebrafish population, which Jale presented to you. This work was funded by R122 Guo, and it was done in 2 labs, and in Dr. Guo's lab in UCSF and Dr. Palmer's lab in UCSD. Thank you.